So we have two sulfur containing amino acids, that is cysteine, cysteine, cysteine and methionine. In cysteine, sulfur is attached to one carbon, and in methionine, sulfur is attached to two carbons. Valency of sulfur is 2. That's why in case of methionine, both the valencies are already occupied, but in case of cysteine, we have space for one hydrogen, so that it is a very special sulfhydryl group present in cysteine. Because of this sulfhydryl group, cysteine is polar, but methionine is not having any sulfhydryl group. That's why methionine is non-polar. And in case of methionine, the sulfur is attached to two carbons, which is a very strong bond, very difficult to be made in body. That's why we cannot make methionine in body. So I will say methionine is essential. We have to take it in diet. But in body, methionine can be converted to cysteine, and therefore I will say cysteine is non-essential amino acid, right? And I already told you in gluconeogenesis topic that both these amino acids are coming under glucogenic amino acid category. You know there are 18 glucogenic amino acids. So cysteine and methionine are also in this category, glucogenic amino acid. So now let's make methionine homocysteine cycle. So I am saying methionine is essential. So from diet we are getting methionine, right? Then methionine gets converted to S-adenosyl methionine. S-adenosyl methionine. You can see here the name is very clear that we are adding adenosyl group here, and on the sulfur adenosyl group is attached here. That's why the name S-adenosyl methionine. And to make the S-adenosyl methionine, the enzyme required is methionine adenosyl transferase. Methionine adenosyl transferase, that is short form MAT. MAT type 1 and 3 are in liver, and MAT type 2 is in extrahepatic tissues. Now let's continue with this SAM. This SAM will donate methyl group, and when SAM will donate methyl group, it will be converted to S-adenosyl homocysteine, and here any acceptor in body can take this methyl group, forming methyl acceptor. There are many reactions which require methyl group from SAM, and these reactions I am going to write here. One is the 7-methyl guanosine cap of mRNA. 7-methyl guanosine cap of mRNA. Second is norepinephrine to epinephrine conversion. Norepinephrine to epinephrine conversion requires SAM. Next is syphilin to lecithin. Syphilin, which contains the nitrogenous base ethanolamine, that is converted to lecithin, which is a phospholipid. Syphilin, lecithin, both are phospholipids, and lecithin is having the nitrogenous base choline, and here in this reaction, from the conversion of ethanolamine to choline, we require 3 methyl groups to be added. They are also given by SAM. Then fourth is in the formation of creatine from guanidoacetate. Creatine is formed. Here also methyl group is added by SAM. And the last and the fifth is acetyl serotonin. Acetyl serotonin will form melatonin. So these 5 reactions in body requires methyl group from SAM, right? Now let's continue in the word cycle. S-adenosyl homocysteine will lose this adenosyl group and will be converted to homocysteine. Homocysteine is a dangerous compound in body. It is causing dangerous vascular diseases if accumulated in the body. So in a normal body, this substance is not allowed to get accumulated. So there are 2 ways of metabolism of this dangerous compound homocysteine. One way is that if enough of methionine is available in diet, then homocysteine will be converted to cystathionine. But if enough of methionine is not available in diet, then this homocysteine will be used to make methionine, right? So let's take this route first, that homocysteine to methionine conversion. So here we have to add which group? Here we have to add methyl group, because from S-adenosyl methionine to S-adenosyl homocysteine conversion, we have removed methyl group. So now from homocysteine to methionine conversion, we have to add this methyl group, and the name of enzyme is homocysteine methyl transferase. This enzyme requires methyl group. So who will give this methyl group? Here comes the role of vitamin B12, that is cobalamin. So we have methylcobalamin, which will give this methyl group here, gets converted to cobalamin, and now we have to again replenish this methylcobalamin by adding methyl group again. Now here, who is the methyl donor? Here comes the role of tetrahydrofolate, and we have N10 methyl tetrahydrofolate, which is used here, giving rise to tetrahydrofolate. So this methyl group is given to B12, and finally it is given to homocysteine for the formation of methionine. Right? Now look here, in B12 deficiency, what will happen? In B12 deficiency, this step will not occur, that is, N-methyl tetrahydrofolate is not converted to tetrahydrofolate. So the accumulation of methyl tetrahydrofolate occurs in the body. This is known as folate trap. So look here, folate trap. Folate trap is not because of deficiency in folate. Folate is vitamin B9. So it is not deficiency of B9. It is deficiency of vitamin B12 which leads to folate trap. That is, there is too much of folate in the body, and that folate is in the form of methyl tetrahydrofolate, because this reaction of methyl tetrahydrofolate to tetrahydrofolate is not allowed. So methyl tetrahydrofolate accumulates. Let me tell you, this methyl tetrahydrofolate has only and only one role in the body, and that is this reaction, only this reaction which we are discussing. But this tetrahydrofolate has so many functions in the body, and now this tetrahydrofolate is not available. So although there is too much folate in the body in the form of methyl tetrahydrofolate, but this folate is unable to do the function, because the functional form is tetrahydrofolate, which is not available. Therefore this is also called functional deficiency of folate. There is too much folate in the body, but this folate is of no use, and this folate trap, that is, all the folate gets trapped in the form of methyl tetrahydrofolate. This occurs in B12 deficiency, not in B9 deficiency, right? Be careful with this point. So this was one route of the metabolism of homocysteine, that is, homocysteine converted to methionine. Now let's see the other route, that is homocysteine to cystathionine formation. Here the name of enzyme is cystathionine synthase. You know that whenever I write synthase means ATP is not used, right? If I write synthetase means ATP will be used. Okay. Now this enzyme cystathionine synthase requires vitamin B6. So you tell me, in B6 deficiency what will happen? In B6 deficiency, this step will not occur, so homocysteine will accumulate in blood, right? In B12 deficiency also this reaction will not occur, so again homocysteine will accumulate, If B9 deficiency, that is folate deficiency, folate is deficient, this step will not occur, so again homocysteine will accumulate. So homocysteine accumulation occurs in case of B6, B9 or B12 deficiency, right? So let's see this topic, homocystinuria. Let me tell you, in blood what is increased in this case is homocysteine. Look here, I have written cysteine, but in urine which compound is coming is homocysteine. Homocysteine is coming in urine. Now I have written cysteine. You know that, like, two molecules of cysteine join to make cysteine. Similarly, two molecules of homocysteine, which is having sulfhydryl group, two molecules will join to make homocysteine, which is having disulfide bond in it. So that is homocysteinuria actually. In homocysteinuria, homocysteine is there in urine, homocysteine is there in blood. And I told you this homocysteine is a very dangerous compound. How it is dangerous? Because it will mainly irritate the endothelium of blood vessels. It will irritate the endothelium of blood vessels, leading to dangerous vascular diseases like stroke, atherosclerosis, pulmonary embolism, myocardial infarction. These kind of clinical features are coming in homocystinuria. Other clinical features are mental retardation, ectopia lentis. These patients are also having seizures, osteoporosis, and one term used is Marfanoid habitus. Marfanoid habitus means clinical features or picture is almost similar to Marfanoid syndrome. You know, Marfanoid syndrome is a connective tissue disorder where these patients have typical tall and slim structure with long extremities, like long arms are there. This kind of picture Marfan syndrome is having, which is a defect in elastin, a fibrous protein, right? Now when I say Marfanoid habitus, means the clinical picture is the features of the patient is similar to Marfanoid syndrome, but there's one difference between Marfan syndrome and this Marfanoid habitus of homocystinuria, that in homocystinuria the ectopia lentis which is there is that lens subluxation, that is lens is dislocated downwards and inwards. But in case of Marfan syndrome, which is affecting the fibrous protein elastin, here the lens is dislocated outwards and upwards, right? So this is the main difference. Now coming to the types of homocystinuria. I will divide it into two categories: genetic, which is because of some enzyme deficiency. On the other side, it can be acquired, and it can be acquired because of vitamin deficiency. You know which vitamins? Vitamin B6, B9 and B12 deficiency. And can you relate here that vitamin deficiency is leading to atherosclerosis and stroke kind of things? Vitamin deficiency is leading to this. So one clinical point here, that whenever you see a young patient with MI or stroke, always get his homocysteine levels checked, right? Now one genetic homocystinuria is left, and I'll be just taking it after I tell you the synthesis of cysteine. So look here, I was telling you this root of homocysteine. Homocysteine is converted to cystathionine with the help of enzyme cystathionine beta synthase is the name of enzyme, and I told you this enzyme requires vitamin B6 and also it requires one amino acid serine, right? After this cystathionine formation, this cystathionine is broken by enzyme cystathionase. This enzyme also requires vitamin B6 and it will break down this cystathionine into cysteine and homoserine. So this way cysteine is getting formed from homocysteine. I told you that methionine in body can form cysteine. So how? Look here, methionine is first forming homocysteine and homocysteine can be converted to cysteine. So can I say cysteine is non-essential, but cysteine becomes essential only if methionine is not available in diet, right? And for the formation of this cysteine amino acid, we require two amino acids, that is serine also we require, methionine also we require, two amino acids are required, and one vitamin, that is vitamin B6 is required, right? Now let's take this topic, genetic homocystinuria. I'm just writing HCU for homocystinuria. This is further of two types, type 1 and type 2. Type 1 is also known as typical or classical type, and type 2 is non-classical type of homocystinuria. In type 1, the enzyme deficient is cystathionine beta synthase. You know, this enzyme is converting homocysteine to cysteine, right? This enzyme is deficient. And in non-classical homocystinuria, various enzymes may be defective, and here the main defect is in conversion of homocysteine to methionine. There may be defect in the formation of active folate, or maybe defect in the formation of active form of vitamin B12, or deficiency of enzyme homocysteine methyltransferase. That is, in short, the defect is in the formation of homocysteine to methionine, and in type 1 the defect is in the formation of cysteine from homocysteine, right? Now this type 1 is more common as compared to type 2. This is more common, and can I say in this case methionine will be increased? Methionine will be increased because excess of homocysteine will be taken up for the formation of methionine, but in this case cysteine is not getting formed, so the cysteine amino acid becomes essential. We have to give cysteine to these patients, right? And the treatment of this patient is that we have to decrease methionine in diet of these patients and give cysteine to these patients, as now cysteine amino acid becomes essential. Now these patients cannot make cysteine in their bodies, right? But in case of type 2 patients, now methionine is decreased because there is a defect in the formation of methionine, and cysteine can be formed in body, so cysteine is non-essential in these patients. So here you can say methionine is decreased but cysteine is normal. And what is the treatment given to these patients? We can give betaine to these patients. Betaine is trimethylglycine. As this betaine is having 3 methyl groups, so it can donate methyl group in the conversion of homocysteine to methionine, and in these patients this is a problem that there's a defect in the formation of methionine from homocysteine, right? So that thing is taken care in the treatment, right? Lab diagnosis of these patients is cyanide nitroprusside test is positive. Cyanide nitroprusside test is positive. Let's see the detail. What is this test? The cyanide nitroprusside test, short form I'm using CNT test. This test is positive for cysteine, homocysteine. In this test, cyanide will break the disulfide bond. It will break the disulfide bond. If cysteine and homocysteine is used, then disulfide bond will be present, that is broken by cyanide, and the liberated sulfhydryl group. Now sodium nitroprusside will react with this liberated sulfhydryl group, giving positive test, and the positive test here is magenta color or a red purple color is the positive test in this case, right? So these 4 compounds having sulfhydryl group also and having disulfide bond also containing compounds will give this test positive, that is cyanide nitroprusside test, right? So now we have to see other disorders of sulfur containing amino acids. So we are done with one, that is homocysteinuria, which is autosomal recessive, and here CNT test is positive, that is cyanide nitroprusside test is positive. The next one I'm going to take is cysteinuria. Cysteinuria is also autosomal recessive, and here also cyanide nitroprusside test is positive. The cysteinuria is a defect in dibasic amino acid transporter. So all the dibasic amino acids, they will be coming in urine in these patients. I'll be taking this in detail after this. Now the next disorder of sulfur containing amino acid is cystinosis. Cystinosis is a generalized lysosomal storage disease. Here also cyanide nitroprusside test is positive. And there's one more disease here, that is cystathionuria. Here cyanide nitroprusside test is negative. So in these 3 diseases, cyanide nitroprusside test is positive, but in cystathionuria it is negative test. Let's take these 3 disorders one by one in detail. So first I'm going to take is this one, cysteinuria. Cysteinuria, I told you it is a defect in the dibasic amino acid transporter. This transporter is present in intestine also and kidney also. So there is defective absorption of these amino acids also, and there's defective reabsorption also of these amino acids. And here these amino acids are coming in urine. These amino acids, you know, COLA, C-O-L-A, these 4 amino acids coming in urine, that is, C stands for cysteine. See, I have not written cysteine, I have written cysteine because in the urine cysteine will be there. O stands for ornithine, L stands for lysine, A stands for arginine. This transporter, these 4 amino acids, frequently used mnemonic here is COLA. C for cysteine, O for ornithine, L for lysine, R for arginine. These 4 amino acids are coming in urine, right? Clinical features, these patients will be having cysteine stones. So they mainly present with flank pain, hematuria because of these cysteine stones present in kidneys. You also know that cysteinuria is one of the Garrod's tetrad. You know, Garrod's tetrad. There are 4 diseases here. These inborn errors of metabolism were studied by the scientist, and these 4 are therefore put under one category. So Garrod's tetrad is, one is cysteinuria, alkaptonuria, albinism, and pentosuria. These 4 diseases come under Garrod's tetrad. Now coming to the treatment of cysteinuria, the main part of treatment is to increase the solubility of these cysteine stones, and how it can be done. One is with good hydration we can increase the solubility. Other is alkalinization of urine. And third is chelating agents like penicillamine can be given. This penicillamine will bind to cysteine and will form a complex here which is soluble in water and can be easily excreted through urine, right? Penicillamine is a chelating agent which will chelate many heavy metals like mercury, lead, iron, and will make a soluble complex which can be excreted through urine. So this kind of chelating agent penicillamine can be used here in cysteinuria, right? Now coming to cystinosis. Here cyanide nitroprusside test is positive. So the cystinosis is a generalized lysosomal storage disease. There is defect in cysteine transporter in the lysosomes. The cysteine transporter which is defective is known as cystinosin. The name of the transporter is cystinosin, and because of this cysteine deposits occurs in various tissues, and the tissues of body which are affected here is bone marrow, cornea, liver, kidneys, etc. Many organs are affected here because I told you it is a generalized lysosomal storage disease. In treatment part, we can give cysteamine. Cysteamine can be given, which will form complex with cysteine, and there is no role of penicillamine in this disease. No role of penicillamine, but cysteamine is the treatment which can be given to these patients, right? Now the next one is cystathionuria. Here cyanide nitroprusside test is negative, and here it is deficiency of enzyme cystathionase deficiency occurs. So cystathionine is accumulated, not homocysteine or homocysteine is accumulated. That's why cyanide nitroprusside test is negative in these patients. This is it with sulfur containing amino acid metabolism. I have taken this topic in detail. Thanks for watching. Best wishes.